Now we're going to move on to how viruses multiply. There are 5 steps in the viral multiplication cycle. So the first step is the virus has to attach to the host cell, also called absorption, and then it has to get inside the host cell. And once inside the host cell, biosynthesis occurs where the virus will make new virus parts, so more viral nucleic acid and protein for the capsid. And then maturation is when the virus is putting together all the viral parts, and then the virus gets released. And at the bottom here, there's a link to a really nice animation of the life cycle of the influenza virus if you want to check that out. So here's just an overview of the viral multiplication cycle with the 5 steps. So you can see the virus, here we have a bacteriophage attaching to the bacterial cell, and then it's going to enter inside the cell. Biosynthesis is when the virus is making new viral parts, so more nucleic acid and viral protein for the capsid. Maturation is when the viral pieces all get put together to make new virus, and then release of the virus from the cell. So the first step of the viral multiplication cycle is called attachment or absorption. So here you can see a bacteriophage binding to a bacterial cell, and it's going to bind to very specific receptors on the host cell. And if the cell does not have the specific receptors that the virus needs, the virus cannot bind to that cell and will not enter it. So this brings us to viral tropism, which just means that viruses are very specific to what type of cells, tissues, or species they will infect. The reason for this viral tropism is because viruses are very specific to which receptors they will bind to. If the host cell does not have the specific receptors on the cell surface, the virus can't bind and will not get inside and infect the cell. So here's some examples of viral tropism. We can have it at the cell level. So for example, on the bottom left, HIV virus will bind to some of our white blood cells because they have the specific receptors that HIV needs to bind to get inside of them. Whereas neurons do not have the right receptors for HIV to bind to, so HIV will not infect neurons. We also have tissue tropism, where viruses will infect a specific type of tissue and not others. It's like influenza virus will infect respiratory tissue because it has the right receptors for the flu virus to get inside. But brain tissue, those cells do not have the right receptors on them, so flu virus can't bind to them and won't infect the brain. And a lot of viruses are very specific to which host or hosts that they will infect. So for example, the myxoma virus will bind to receptors found on rabbit cells, but humans, our cells do not have the right receptors, so we will not be infected by the myxoma virus. Okay, the second step in viral multiplication cycle is viral entry. So either the entire capsid with the nucleic acid inside will enter the host cell, or just the nucleic acid will enter into the cell. So it just kind of varies between whether it's an enveloped or non-enveloped virus. There are 3 methods for viruses to get inside of the host cell once they bind to the specific receptors. So I want you to memorize these 3 different methods of entry into the host cell. So the first one is that if the virus is enveloped, the viral envelope can fuse with the host cell membrane, and then that will allow the viral capsid to enter inside the cell. So the type of viruses that can do this first method of entry would be envelope viruses. So envelope viruses, they do have a viral envelope that can fuse with the host cell membrane, and then that will allow the capsid to enter. The second method of viral entry into the host cell is receptor-mediated endocytosis. So both enveloped and non-enveloped viruses can do receptor-mediated endocytosis and enter that way. So they will bind to receptors on the host cell and trick the cell into thinking that it's something else, and the cell will then bring it in by endocytosis. And the third method of entry is shown in this animation over here, where we have a non-envelope virus. In this case, we have a bacteriophage that is actually injecting its nucleic acid inside the host cell, which happens to be a bacterial cell. So the capsid will stay on the outside and just the nucleic acid will enter with that. So that only happens with non-envelope viruses. So here's just some pictures of two of the methods of viral entry into the host cell. So the picture up here on the top, we have an envelope virus, and you can see that the spike proteins are binding to specific receptors on the host cell membrane. And once the virus binds to the host cell, the viral envelope just fuses right with the plasma membrane, and then that allows the capsid to enter inside the cell. The second picture is showing again an envelope virus binding to specific receptors, and in this case, instead of the viral envelope fusing with the host cell membrane, the virus is going to enter by receptor-mediated endocytosis. So the virus binds to these receptors that normally will bring in something else, like maybe in our cells, we do receptor-mediated endocytosis to bring in like cholesterol, for example. So these viruses will bind to those receptors, trick the cell into thinking it's something else, and the cell will do receptor-mediated endocytosis. So it will grab its plasma membrane and surround the virus, pinch off, and then bring the virus inside the cell. And then the last picture is also showing receptor-mediated endocytosis, and this is happening with a non-enveloped or naked virus, so there's no envelope on this one. So the protein capsid will bind to the specific receptors on the cell, trick it into thinking it's something else, and then that will trigger receptor-mediated endocytosis. The cell will reach out with its membrane, surround the virus, pinch off, and then bring the virus inside the host cell. Okay, and then here's just some electron microscope views. This is just showing you an envelope virus here, and it's going to be entering in by receptor-mediated endocytosis. So you can see the cell is reaching out its membrane and surrounding that virus, and we'll bring it in by endocytosis. And then the picture on the right is a picture of a herpes virus, also an envelope virus, and this one is going to be entering by fusing the viral envelope with the host cell membrane. So that's how it enters. Okay, the third step of the viral multiplication cycle is biosynthesis. So once the virus is inside the cell, it needs to make new virus. So remember that viruses are only made out of nucleic acid and proteins. So it only has to make two things to make more virus. If the virus is using double-stranded DNA as its genetic material, it's just going to use all of the host cell enzymes because that's what our cells use, double-stranded DNA, and so the viruses are very similar, and so it's very easy for them just to use our enzymes and make more virus. But if the virus is using either double-stranded or single-stranded RNA as its genetic material, it's going to do some things that our cells do not do. So they have to bring in some of their own enzymes with them. Okay, maturation is when the new virus parts are going to be put together. So we're going to assemble the capsid and then put the viral nucleic acid inside there. And inside the cell, you can oftentimes actually see the little virus factories where they're actually making all of the new virus and putting those pieces together. So maturation, we're just going to put together the new viral parts, and they actually will just self-assemble, which is pretty cool. Okay, and this is showing you a bacteriophage, and you can see here on the bottom right, you can see the self-assembly going on with maturation phase. So all of these pieces that are made inside the host cell, they just automatically just self-assemble and snap together. and make new virus. The last step of the viral multiplication cycle is to release the newly made virus from the host cell. So non-envelope viruses will lyse the host cell. So in this animation on the bottom here, we've got some non-envelope bacteriophages, and when they're ready to be released, they just lyse the bacterial cell. Envelope viruses are very different. They're going to be released from the host cell by budding. So on the bottom left here, we've got a picture of a white blood cell. And the virus that is budding off here and being released, that's the HIV virus. So remember that these envelope viruses, they get their envelope from the host cell. So as the HIV virus leaves the white blood cell, it takes part of the host cell membrane with it. So the host cell does not die immediately because you're not lysing it and exploding the cell all at once, but eventually it will die because it's making all this virus and just slowly releasing those, and eventually the host cell will die. Here's just a picture of a bacterial cell, and you can see that it's been infected with lots of bacteriophages. You can see the protein capsid that was left on the outside here. They entered by injecting their nucleic acid inside the bacteria. That directed the bacterial cell to make lots of new bacteriophages, and then once the viruses were all assembled, then they just lysed the bacterial cell and then released a bunch of new bacteriophages. This picture is showing the release of envelope viruses by budding from the host cell. So if you start up here at the top, you can see the host cell membrane here, and the virus has made all of the spike proteins and has already put them into the host cell membrane. So when the virus goes to leave, you can see the capsid here in blue. It has nucleic acid inside. So when it leaves the host cell, it just buds off and just takes part of the host cell membrane with it. So here's a picture of the viral growth curve. What we have is the log number of viruses that's on the y-axis here, and then we've got time on the bottom. So step number one is just showing you that we've added the virus to the cell culture. And then the eclipse phase, you can see that the number of viruses has really decreased and they've disappeared. So what's happening in this eclipse phase, why there's no detectable viruses outside of the cells, is because they're all inside the cells. So during the eclipse phase of the viral growth curve, the reason that there's no viruses outside the cells is because they're all inside. And the two steps of the viral life cycle that are occurring during the eclipse phase would be the biosynthesis phase and then the maturation phase. So they're busy making new viruses in the biosynthesis phase, and then the maturation phase, they're self-assembling all the virus pieces and getting ready to release. And then the burst phase, you can see the number of viruses outside the cells really increases, and this is where the virus is being released from the host cell. Okay, remember we talked about there were two different types of bacteriophages. We had the virulent phages, which only do the lytic cycle, and then we have the temperate phages, which do the lysogenic cycle and then the lytic cycle later. Okay, and then we'll just talk about these terms in the next picture. So this picture shows the life cycle of the virulent and temperate bacteriophages. So remember that the virulent bacteriophages can only do the lytic cycle. So they'll attach to the bacterial cell, inject their viral nucleic acid inside the bacteria, and then they immediately will start making new virus, and then they will release the virus by lysing the cells. So they can only do that lytic cycle. But the temperate bacteriophages, remember temperate means moderate, so they will first do the lysogenic cycle. So they will inject their nucleic acid inside the bacterial cell, and then they insert it right into the bacterial chromosome. So that viral DNA is called the prophage. And then the bacterial cell that contains the prophage, the bacterial cell is called the lysogen. So the bacterial cell is the lysogen, and then the viral DNA is the prophage. And when the bacteria divides, it copies the prophage, passes that on to the daughter cells, and then if the bacterial cell gets into a bad environment, the temperate phage actually senses that and will cut itself out of the bacterial chromosome and then enter into the lytic cycle and start making new virus and then go find another bacterial cell that might be in a better environment. There are two very important results of lysogeny, of doing this lysogenic cycle with the temperate bacteriophages. The first one is called phage conversion. So the first important result of lysogeny is called phage conversion. So what this means is that there are some bacteria that if they are not infected with the bacteriophage, they do not make you sick. But if they are infected with the bacteriophage, they do make you sick. So a great example of phage conversion is with the bacteria that cause the disease diphtheria. So the Corynebacterium that cause diphtheria, if they're not infected with the bacteriophage, they don't make you sick. But if they are infected with the phage, then they do cause diphtheria. So how they make you sick by being infected with the phage is that the virus contains genes that make toxins that cause the signs of diphtheria. So that's what makes you sick. Besides phage conversion, the other important result of lysogeny is that it allows specialized transduction to occur. And remember, transduction is one of our forms of horizontal gene transfer. And here's just a picture of specialized transduction that happens with the temperate bacteriophages in the lysogenic cycle. So when the prophage cuts itself out of the bacterial chromosome, sometimes it's a little bit sloppy and cuts out a bacterial gene as well. So when the virus makes new virus, it packages up the bacterial gene along with the viral genes, and then when the virus infects the next bacterial cell, it injects and transfers that gene from the other bacteria to this new bacterial cell. And so now this different bacterial cell has a new gene that it didn't have before, and that'll cause genetic variation. So remember, that is a form of horizontal gene transfer. So that's transduction. So using a virus to transfer genes from one bacterial cell to the next.